Hello everyone, welcome to Medical Guruji. Today is our third week lecture of course where we will explain the spectrum of activity of antibiotics. In the last lecture we discussed about the mechanism of action of various antibiotics in details. Today we will understand how different antibiotic target specific bacterial population which is crucial for effective treatment and responsible for antimicrobial stewardship also. In this lecture, we will examine mainly the two categories of antibiotics based on their spectrum of activity, narrow spectrum and broad spectrum antibiotic. We will also discuss their mechanism of action, examples, advantages and their clinical applications. So starting with the narrow spectrum antibiotics. Narrow spectrum antibiotics are the antimicrobial agents which are effective against a limited range of bacteria. They target specific type of organisms such as gram-positive or gram-negative bacteria rather than a broad array of pathogens. Their focused activity minimizes disruption to the body's natural microbiota. Mechanism of action of narrow spectrum antibiotics. They act by targeting specific bacterial processes or structures that are unique to bacteria they are designed to combat. Their mechanism may include inhibit cell wall synthesis. For example, penicillin G for gram-positive bacteria is a cell wall synthesis inhibitor. Blocking protein synthesis by binding to the bacterial ribosome. And third is the interrupting nucleic acid synthesis or function. Here you can recall the last lecture, that is week 2 lecture, where we discuss all the three mechanisms in details. Examples of the narrow spectrum antibiotics are penicillin G and penicillin V. These are effective primarily against gram-positive bacteria such as Streptococcus species. They inhibit bacterial cell wall synthesis by targeting penicillin binding proteins PBPs. So second example is vancomycin. It is highly specific for gram-positive bacteria including Staphylococcus aureus and Clostridioides difficile. It inhibits the peptidoglycan synthesis in the bacterial cell wall. Third example is isoniazid. It targets the Mycobacterium tuberculosis by inhibit mycolic acid synthesis, which is essential for the bacterial cell wall. Fourth example is polymyxins, that is polymyxin B or polymyxin E, that also known as colistin. It is specific for gram-negative bacteria and act by disrupting the outer membrane, that is lipopolysaccharides. It is used for resistant infection caused by bacteria like Pseudomonas aeruginosa. Advantage of narrow spectrum antibiotics. First, they have less likely to affect beneficial bacteria in the normal microbiota, reducing side effects such as diarrhea or yeast infections. By targeting fewer bacterial species, they reduce the selective pressure of the emergence of drug-resistant strain in the unrelated bacterial populations. Third is the reduced impact on commensal bacteria helps maintain the balance of body microbial environment. Clinical use of narrow spectrum antibiotics. These are often prescribed when the causative organism of an infection has been identified or the goal is to minimize damage to the body normal microbiota. And third is the broad spectrum antibiotics are unnecessary or pose risks such as in uncomplicated infections. Now we will discuss the broad spectrum antibiotics. Broad spectrum antibiotics are antimicrobial agents which are effective against a wide range of bacterial species including both gram-positive and gram-negative bacteria. These drugs are particularly useful when causative organism of an infection is unknown or in case of mixed infection. Mechanism of action of broad spectrum antibiotics. These act on the fundamental processes shared by many types of bacteria such as cell wall synthesis, protein synthesis or nucleic acid synthesis. Their ability to target a wide array of bacteria stems from their mechanisms affecting conserved bacterial structure or enzymes. Examples of mechanisms, as we discussed in the last lecture already in the details. Inhibition of cell wall synthesis like carbapenem, imipenem exhibit a bacterial transpeptidase enzyme which prevent cross-linking of peptidoglycan in both gram-positive and gram-negative bacteria. Second is inhibition of protein synthesis like tetracycline binds to the 30S ribosomal subunit reversibly, which prevent amino acetyl tRNA attachment. Then chloramphenicol binds to the 50S ribosomal subunit and inhibit peptide bond formation. Third example of mechanism is by inhibition of nucleic acid synthesis, that is fluoroquinolones. In this category, that is example is ciprofloxacin, which inhibit the DNA gyrase and topoisomerase 4, affecting DNA replication in broad range of bacteria. Examples of broad spectrum antibiotics are tetracyclines, which are effective against gram-positive and gram-negative bacteria, as well as atypical pathogens like chlamydia, mycoplasma. Second example is chloramphenicol. It covers a wide range of bacteria, including anaerobes, but is used sparingly due to potential toxicity. Third is fluoroquinolones. These are these have broad efficacy against gram-negative and some gram-positive bacteria. Fourth is amoxicillin clavulanic acid, that is augmentin. The combination expands activity of beta-lactamase producing bacteria, covering both gram-positive and gram-negative organisms. Fifth examples are carbapenems. Example imipenem or meropenem. Very broad spectrum activity, including multi-drug resistant organism, though not effective against certain bacteria like MRSA. Advantage of broad spectrum antibiotics are: first is versatility in unknown infection. It is useful in empirical therapy when the causative pathogen has not been identified. Second is the coverage for mixed infection. It is effective against polymicrobial infection involve multiple bacteria. Third is wide applicability. It is used in healthcare setting for critically ill patients. Disadvantage and the risk of broad spectrum antibiotics are: first one is antibiotic resistance, that is overuse contribute the emergence of multi-drug resistant bacteria. Then it can harm the normal flora, leading to the conditions like Clostridioides difficile associated diarrhea. Third is the broad spectrum drugs may cause more side effects such as secondary infections or toxicity. Clinical use of broad spectrum antibiotics, which these are frequently employed for severe or life-threatening infection when immediate treatment is necessary. Polymicrobial infection, example intra-abdominal or soft tissue infections. Situations where narrow spectrum antibiotics are ineffective or inappropriate. Hence, understanding the spectrum of activity of antibiotics involve analyzing the range of bacterial species that a particular antibiotic can target effectively. So, we will discuss the step-by-step approach to grasp this concept. First is learn the classification of antibiotics. Then, as we already discussed, narrow spectrum antibiotics and broad spectrum antibiotics. Then, second is understand the gram staining. So, gram-positive bacteria and gram-negative bacteria. These are two different categories based on the gram staining. Gram-positive bacteria have thick peptidoglycan wall that retain the violet stain. Examples include Staphylococcus, Streptococcus, etc. In the gram-negative bacteria, they have thinner peptidoglycan wall and an outer membrane, making them resistant to certain antibiotics. For example, E. coli and Pseudomonas aeruginosa. Antibiotics effectiveness often depend on whether they can penetrate the cell wall or outer membrane. So, understanding gram staining help in predicting the spectrum. Third approach is by study mechanism of action. As we already discussed in the last lecture, there are 5 main classes based on the mechanism. Cell wall synthesis inhibitor. These are effective mostly against gram-positive bacteria. In this category, mainly penicillins and some like carbapenems also work against gram-negative bacteria. Second class was a protein synthesis inhibitor, which target the ribosomal subunit and have broad spectrum, examples like tetracyclines. Third was nucleic acid synthesis inhibitors. These also have a broad spectrum due to conserved DNA replication enzymes across bacteria. In this category, the fluoroquinolones are involved. Fourth category was metabolic pathway inhibitors. Sulfonamide and trimethoprim are broad spectrum but primarily used against gram-negative and some gram-positive bacteria. Last was cell membrane disruptors, like polymyxins targets gram-negative bacteria due to their action on lipopolysaccharides. Fourth, Fourth approach to understand this concept is consider antibiotic resistance. Resistance mechanisms such as beta-lactamase production or efflux pumps can affect antibiotic spectrum. For example, penicillin may lose effectiveness against certain gram-positive bacteria producing beta-lactamase, while carbapenems can overcome many resistance mechanisms. Fifth approach is use clinical context. In this, there are two therapies like empirical therapy and definitive therapy. Empirical therapy is broad spectrum antibiotics are preferred when the causative pathogen is not known, and the definitive therapy is given when the in definitive therapy narrow spectrum antibiotics are used once the pathogen is identified to minimize the microbiota disruption and resistance risk. Sixth approach is refer to the antimicrobial charts. Consult resources such as antibiotic susceptibility tables or charts showing the spectrum of activity for specific drugs. These tools provide detailed insight into which bacteria each antibiotic is effective against. Seventh concept is understand pharmacokinetics and pharmacodynamics. Some antibiotics are more effective against certain bacteria due to their ability to reach the site of infection, pH sensitivity, and other factors. For example, polymyxin work well in case of gram-negative sepsis because they penetrate the outer membrane. Macrolides, while broad spectrum, may be preferred for respiratory infection because uh, respiratory infection caused by the atypical bacteria like mycoplasma. The clinical implication by clinical implication of the spectrum of activity of antibiotics like helps in the deciding the targeted therapy, that is using narrow spectrum antibiotic when the causative agent is known. It helps minimize the impact of on beneficial bacteria and reduce the risk of antibiotic resistance. Then empirical therapy when the causative agent is unknown. Broad spectrum antibiotics are often used initially. Once the pathogen is identified, therapy can be adjusted to more targeted antibiotics. Then focus on the antibiotic stewardship. Prudent use of antibiotic guided by their spectrum of activity help in combat the growing problem of antibiotic resistance. To summarize the today's lecture, we discussed the narrow spectrum antibiotics that target a limited group of bacteria such as gram-positive or gram-negative organism. They are precise, reducing disruption to normal microbiota, and example include penicillin G, vancomycin, etc. While the broad spectrum antibiotic, these act against a variety of bacterial types, including both gram-positive and gram-negative as well as atypical organisms. They are useful for treating unknown infections but can disrupt beneficial bacteria. Example include tetracycline and fluoroquinolones. This was all about today's lecture on the spectrum of activity of antibiotics. In the next week, we will discuss the pharmacokinetic and pharmacodynamic aspect of antibiotics. If you have an query, kindly comment in the comment box. Do like, share and subscribe our channel. Thank you.